Now let's talk about facilitated diffusion. What actually is diffusion? It's again the movement of molecules from a region of high concentration to a region of low concentration. It is occurring along the concentration gradient. So obviously it will be a passive process. There is no requirement of any energy. But here they said facilitated. Facilitated means something is assisting this process of diffusion. Someone is facilitating the process of diffusion. So the proteins which help in helping the diffusion process, which play a role in diffusion to take place, they are called channel proteins or carrier proteins. Okay, now why there is a requirement for proteins to assist this process? Now consider, now this is your plasma membrane. Plasma membrane is a phospholipid bilayer. Basically, that means interior it is made up of lipids and onto the exterior it has proteins. Okay. So basically they are made up of lipids, where this is hydrophobic end, hydrophilic, I'm sorry, hydrophilic, hydrophilic head, and these are the hydrophobic tails. What do I mean by saying hydrophilic head and hydrophobic tail? That means hydrophilic head, these are water loving. They are basically, they allow any polar molecule to enter through them. Hydrophobic, that means these are water hating or fat loving. Now for example, if I want water molecules to enter through the membrane, these are hydrophilic heads, that means they allow the water molecule to pass through them. But this region, which is made up of lipids, fatty acids, it does not allow the water to enter through it. That's the reason we require certain channel proteins. Aquaporins are the channel proteins that allow water to pass through the membrane. So here the movement is from a region of high concentration to a region of low concentration, which is assisted by proteins, but there is no need of any energy. Okay, that's the reason it is called facilitated diffusion. Now this facilitated diffusion, here the movement through the protein can be uniport, symport or antiport. That means, for example, this is my channel protein. If it involves the movement of a single solute molecule either into the cell or outside the cell, either means it can be either into the cell or outside the cell, then it is uniport. When I say symport, that means when two solute molecules are moving together simultaneously into the cell, that is called symport. What is antiport? Where one molecule is moving inside, other molecule is moving outside, that is called antiport. So these type of movements are usually seen in case of facilitated diffusion, and we say that it is uniport, symport or antiport. Now if you see the water molecules moving through the aquaporin, that is uniport, where the molecules are just entering, it is one-way flow. Okay, now let's talk about active transport or uphill transport. Till now we have seen processes where there is movement of molecules occurring from a region of high concentration to a region of low concentration, where there is no requirement of energy. Just imagine there is a point from A to B and you are standing somewhere here. If I tell you to climb from A to B, obviously we require more energy than when we come down the hill, right? So that's the reason active transport is also called uphill transport, where we require more energy. Why is it so? This is because here the molecules move from a region of low solvent concentration to a region of high solvent concentration. Okay, that means if there is, consider its plasma membrane, okay, and there are solute molecules present. And water is present, okay, and here also there are solute molecules, but they are very less, and water is present. Now what actually happens is the movement of water takes place from region of low solvent concentration to a region of high solvent concentration. That means it is occurring from low to high against the concentration gradient. Okay, this is a point that you need to remember. That's the reason, as it is occurring from low solvent concentration to high solvent concentration, energy is required. This energy is in the form of ATP, which is adenosine triphosphate. Adenosine triphosphate. Okay, and the proteins which assist this process. See, molecules cannot move against the membrane freely. Okay, they can be polar molecules also, as I have discussed earlier in the previous slide. The inner membrane of the plasma membrane is completely made of lipids. It does not allow water molecule or ions to pass through it freely. Therefore, we require proteins, but the proteins here are called pumps. Why? They are pumping the ions against the concentration gradient. So this is called active transport or uphill transport. Now let's talk about osmosis. What is osmosis? It is movement of solvent molecules from a region of high solvent concentration to a region of low solvent concentration through a semi-permeable membrane. If you remember, in the beginning of this lecture, I told you what actually is a semi-permeable membrane. A membrane which only allows solvent molecules through it and does not allow only solute particles. So during osmosis, can solute molecules move? Obviously no. Let us consider a beaker. Okay, and this beaker is separated by egg membrane, which is a semi-permeable membrane. Okay, and let us consider there are water molecules here and very less solute molecules. Consider here also water. Okay, and it has many solute particles. Now if you see here, which region has more water potential? Obviously this is the region which has more water potential. This has very less water potential. So what is osmosis? It is movement of solvent. Are the solute molecules moving? No. It is the water which is moving from this portion A to portion B. Okay, till equilibrium is maintained. That means till both the concentration in both the chambers A and B is equal. Okay, that is called osmosis, where there is movement of solvent molecules from a region of high solvent concentration to a region of low solvent concentration through a semi-permeable membrane. What do I mean by saying solvent? Solvent can be water. Okay, when I say solute, it can be glucose, it can be salt, it can be sugar molecules, etc. Okay, and again this is a passive process. Why? The movement is taking place from a region of high water potential to a region of low water potential. So it is along the concentration gradient and not against the concentration gradient. What is osmotic pressure? It is the pressure required to stop the movement of water completely. For example, if I take a suction pump or something and put pressure, then what happens? Water starts moving from this side. So the amount of pressure that we apply to stop the overall movement, net movement, is only known as osmotic pressure. And we have discussed the examples: egg membrane, gourd bladder, parchment paper. These are examples of semi-permeable membranes. In osmosis, it is very important to mention that movement of solvent molecules takes place from a region of high solvent concentration to low solvent concentration through a semi-permeable membrane. This is very important. Okay. Hypertonic, isotonic, hypotonic solution. Hypertonic solution. When will I say that a solution is hypertonic? For example, consider there is a beaker with 100 ml of water and 500 grams of sugar in it. Consider another beaker which again has same 100 ml of water, 100 ml, and I added 5 grams of sugar. Now let us put a plant cell or any animal cell, it can be liver cell, red blood cell, anything, into both beakers A and B. And let us see what actually happens. Now see here, this beaker A is having 100 ml of water and 500 grams of sugar. Obviously it is highly concentrated. Water potential is very less when compared to the plant cell or animal cell. So what actually happens? Water which is present inside the cell starts moving outside. As a result, what happens? The cell starts shrinking. This is what we observe when a cell is placed in a solution having higher concentration than itself. So here the cell is placed in a solution which is having more concentration, more solute particles in it than itself. So water potential of the cell is more compared to the water potential of the beaker. As a result, the water is moving out of the cell into the beaker and the cell can shrink. Now consider another case I told you where I'm adding 100 ml of water in 5 grams of sugar and 5 grams of sugar. Here what happens is the water potential of the cell is high compared to that present in the cell. As a result, water starts moving inside the cell. Okay? And this can result in swelling. If it is an animal cell, it can result in lysis. That means the cell can break, or it can result in swelling. I'll tell you why in animal cells it can break, and in plant cells why it results in a swelling. Okay. Now consider a case where the concentration, the water potential of the cell is equal to the water potential in the beaker. Then there will be no net movement. What do I mean by saying no net movement? That means if two water molecules are moving inside, then 2 water molecules will come outside, something like that. So there is no net movement. So such a type of solution is isotonic. That means the concentration inside the cell and outside the beaker is same. Hypotonic means the concentration of the solution in the beaker is very less compared to the solute concentration in the cell. Hypertonic, it is the case where the solute concentration is very high in the beaker compared to the cell. So water from the cell starts moving out. If you observe in all these 3 cases, always the movement of water takes place from a region of high water potential to a region of low water potential. Clear? Now let us understand the same mechanism, like how osmosis takes place in animal cells. Important point to remember is animal cells, they lack a cell wall. Okay, the outer boundary in the animal cell is plasma membrane. They do not have a cell wall. So what actually happens is, if you take an animal cell and put in hypotonic solution, what do I mean by saying hypotonic solution? That means water potential is high in the beaker and less in the cell. So what happens? Water starts moving inside. As water starts moving inside, the cell swells and it breaks because there is no rigid cell wall to provide that pressure. So what happens? The cell simply gets lysed. Now let us consider a case where I am putting a cell in hypertonic solution. That means water potential is less in the beaker and high in case of the cell. So what actually happens is, water starts moving from a region of higher water potential to a region of lower water potential. That means it starts moving from the cell to outside. It results in shrinking. Okay, shrinkage occurs. Now this is isotonic, where I said there will be no net movement. See, the amount of water that is getting inside is equal to the amount of water that is moving outside. So such type of solution is isotonic. Important point to remember is, in case of animal cells, when the cells are placed in hypotonic solutions, they break or they lyse. Okay. Now let us understand what happens if plant cells are placed in hypertonic, isotonic and hypotonic solutions. Let us first consider if plant cells are placed in hypotonic solution. That means the water potential is high outside and less inside. Then what happens? Water starts moving inside, and as a result it starts swelling. But here it won't break. Why? This is because plant cells have a rigid cell wall which is made up of cellulose. What does the cellulose do? Obviously, when water enters into the cell, it creates some pressure inside the cell. That pressure is called turgor pressure. What is turgor pressure? It is the pressure which is exerted by water as it is moving inside the cell. But wall which is present, cell wall, it also exerts a pressure which is opposite and equal in magnitude. That allows the cell not to break. That means, for example, now see, water is moving inside the cell. As a result, there is pressure developed. That turgor pressure is 10 Pascal, for example. Now wall also exerts pressure against this turgor pressure in opposite direction. that will be equal, same magnitude, but it is opposite in direction, so we represent it as minus 10 pascals. So this is wall pressure. Because of this wall pressure, the cell is not breaking, okay, it does not lyse. Now such a cell is said to be turgid cell because it is filled with water and it is under pressure, turgid. Okay, if a cell is placed in isotonic, then what happens? The amount of water entering into it and coming out of the cell is equal. In such cases, it is said to be a flaccid cell, okay, where the pressure is zero, pressure potential is zero. Now consider a cell when the plant cell is placed in hypertonic solution. That means water potential outside is very less. So water starts moving out only from the cell. Initially, water moves from the cytoplasm. Okay, consider this normal cell. You have vacuole here and this is the cytoplasm. It starts moving from the cytoplasm first. The water starts moving from the cytoplasm and then from the vacuole. What is incipient plasmolysis? It is the stage when water is being lost from the vacuole and the cytoplasm. And if you continue placing the cell in the hypertonic solution for a long time, then what actually happens is completely the protoplast, that means the cytoplasm along with the vacuole, they do not remain intact with the cell wall. That means they come out of the cell wall, and that stage is called plasmolysis. Okay, what is plasmolysis? It is the stage where completely water is lost and the protoplast gets shrink. This is protoplast, that means the vacuole along with the cytoplasm, they shrink. Okay, when this plasmolysed cell, when it is again placed in hypotonic solution, then what happens? It can become a normal cell. Okay, that process is called deplasmolysis. So it is something which can be reversible. Okay, this plasmolysis process is a reversible process. If you again keep the plasmolysed cell into hypotonic solution, then what happens? Water starts entering into the cell and cell becomes turgid. That process is called deplasmolysis. Okay. Now let us understand what is imbibition. Okay. For imbibition to take place, there must be adsorbent present. Okay, an adsorbent and water potential gradient, that means difference in water potential, is required for imbibition to take place. What is adsorbent? It can be any dried or semi-dried substance. Example, dried seed, wood, fibers, sponge, all these are examples of adsorbents. Now absorption by imbibition is also a type of diffusion. It does not require any energy. Okay, it is also a type of diffusion where movement takes place from a region of high water potential to low water potential. But for imbibition to take place, there must be affinity between the adsorbent and water. Consider the dried seeds. I told you that seeds, when they are under harsh conditions or unfavorable conditions, they develop a seed coat. But for germination to take place, they require some amount of water. So initially these dried seeds, they take up water by process of imbibition. Okay, once they take up some amount of water, slowly there will be development of plumule and radicle that later give rise to the root and shoot system. Okay, so basically initially for germination, the water that has to be taken up by the dried seeds under favorable conditions is by process of imbibition. Now when this water is taken up by the plant, there will be pressure created. That pressure we call it as imbibition pressure. Okay, 1 minute, where my marker? Oh, sorry. That is called imbibition pressure, or it is also represented as matric potential. Usually it is negligible in living plant cells. It is taken into account only in dried seeds. Okay, so matric potential is nothing but the imbibitional pressure which occurs when water enters into this dried seeds. Even see, water is absorbed by cotton fibers. Cotton acts as an absorbent, okay, unlike in rubber sheet. So imbibition is a type of diffusion only, but it occurs in solid colloids. Okay, whereas diffusion, it is more prominent in gases and liquids. We have seen solid getting diffused in liquids, gases getting diffused in gases, or liquids getting diffused in gases, something like that. But imbibition takes place in solid colloid molecules. Now after studying the different processes like osmosis, passive transport, that is diffusion, facilitated diffusion, active transport, imbibition. Now let us talk about the theories of water translocation. That means how actually is water transported. Now just imagine the giant sequoia trees of gymnosperms or the cone plants. How actually is water getting transported from the soil to the roots and from the roots to the tip of the leaves of the plant in such tall trees. So various theories were put forward to explain this movement of water. So the first one is vital force theory. This theory was given by J.C. Bose, prominent scientist. He has given the theory, and according to this theory, he said that water along with mineral nutrients, we know that water is a very good solvent. It is an universal solvent. That means it helps in, it can dissolve any polar solute in it. So basically water along with the mineral nutrients present in the soil, they get dissolved. They are absorbed by the cortical cells of the roots and they pump to the xylem elements. So according to J.C. Bose, he said that, I'll show you this. He said that the cortical cells, these are the cortical cells, right? They take up the water along with the mineral nutrients and they pump it to the xylem elements. Pump means what? Here it is by active transport. That is what he is telling. At the expense of energy, it is taking place. This was the theory which was put forward by J.C. Bose in his vital force theory. The next theory is capillarity. What actually is capillarity? Have you ever seen capillary tubes? These are capillary tubes, very thin tubes. They have very small diameter. Now these capillary tubes, they, for example, if you take a capillary tube and put it in a beaker containing water or any colored solution, what happens is immediately the water from the beaker or any plate starts moving inside the capillary tube. The movement of water into the small narrow space without any external force being applied is only known as capillarity. You are not applying any force, just the narrow diameter is causing the water to lift itself up. How is it happening? That's because of adhesion. That means the water molecules have attractive forces to the walls of the tube. In the same way, the xylem vessels or the xylem tracheids, they are very small in diameter. So this theory explains that movement of water takes place through the small diameter of the tubes by process of capillarity. Smaller the diameter, greater will be the lifting force. But the main drawback of this theory is that though water is moving up through the xylem elements, but the amount of water that travels is very less. So it could not explain it failed to prove that the transportation of water takes place by capillarity, because though the lifting force is greater if the diameter is small, but the amount of water that usually gets in is very less. Okay, so it failed to explain. But the main principle of capillarity is adhesion. That means the water molecules have attractive forces to the tubes of the xylem element, that is causing the water to move up. Okay. So the next theory that was put forward to explain how actually water transportation took place was root pressure. It was proposed by Priestley. Okay, and root pressure is nothing but when water, now see, consider these are the soil particles. We know that the soil particles have a very tight film of water. Can that be used by plants? No. This is the hygroscopic water. But they also have a very loose film of water which can be taken up by plants. That is the capillarity. That is the capillary water. So basically the roots, they take up this water along with the mineral nutrients, and this develops pressure inside the roots. Obviously, I told you when water enters inside, it creates pressure. That is called pressure potential or hydrostatic pressure. Since it's occurring in the root, it is called the root pressure. So this root pressure is positive, and this root pressure is causing the movement of water to take place inside the xylem sap of the roots and from the xylem sap to the xylem stem. This was explained by Priestley in root pressure theory, where he is telling that the water present inside the roots is causing the movement upward against the gravity. Okay, but it was found that the magnitude of this root pressure is very less. It is just 2 bars. So obviously it can't take up the water against the gravity, such a small amount of pressure. So it failed again to prove how the transportation of water is taking place up the plants to the top peak. and this root pressure is absent in conifers. Then how in conifers is the transportation or translocation of water taking place? Okay, so even root pressure theory failed or did not explain how translocation of water is taking place in plants. Now there is an important phenomenon known as guttation. What actually is What actually is guttation? You can see here a beautiful leaf, right? And near the margins of the leaf we can see certain small water droplets. So the loss of water as liquid form in herbaceous plants from the tips of the leaf blade is called guttation. So guttation is loss of water in the form of water droplets. I think you have studied transpiration. Transpiration is the process where water is lost as water vapor, okay, from the stomata, from the aerial parts of the plant. But here the water is lost in the form of liquid droplet from the tips of the leaf blade, and that is called guttation. And guttation is because of excess root pressure. Okay, it is a consequence of root pressure only. Okay. So what was the theory that finally was accepted by many scientists to explain how actually water is getting translocated to the top regions in the plant against the gravity? That was the cohesion tension theory, also known as transpiration pull or ascent of sap, and it was first proposed by Dickson and Joly. Okay. This theory, according to this theory, they say that the main driving force for water to be pulled up from the soil is transpiration. What is transpiration? It is loss of water in the form of water vapor from aerial parts of the plant, basically from the stomata of the leaves, so or the cuticles. So that is called transpiration. So what actually is happening? How is it helping in transport of water from the soil? It takes place in the leaves, right? How does that help in transport? Now consider these are the leaves. We know transpiration is when there is excessive sunlight. Then what happens? Water is lost in the form of vapor into the atmosphere. Okay, the water is lost into the atmosphere in the form of water vapor. Now this region is deficit of water. Then what happens? Water which is present in the petioles of the leaf start moving into this region. Now this region becomes deficit, means less amount of water is present in the petiole region. Then the stem starts giving it water. Now stem is deficit of water as it has given water to the petiole region. So what happens? The stem starts taking water from the root. Means root. So now root becomes deficit of water. It starts pulling water from the soil. We know that soil is having more water potential, higher water potential compared to the roots, obviously. So that's the reason soil takes up water, and this water again is taken up by stem, because the stem is deficit because it has given the water to petioles. Petioles are deficit of water because it has given water to the leaves. Leaves are deficit of water because of transpiration. So this transpiration is causing the water to be pulled from down. It is causing the force, okay, driving force. That's the reason this theory is also called transpiration pull. So basically, one thing you need to now, why is it called transpiration pull? That's because transpiration is occurring at the leaf surface and it is causing the pulling of water from the roots. From like the water is lost from the leaves, it is causing the pulling of water from the xylem of the stem. From the xylem of the stem, it is causing pulling of water from the xylem of the root. The xylem of the root starts pulling water from the soil. So this pulling, what is the driving force for this pulling? Transpiration. Okay. Now this transpiration pull, this theory is also called cohesion tension theory. Why is it so? Now see, transpiration, it is pulling water. That means water is lost as the form of water vapor from the leaves. This causes tension in this region where water is lost. As a result of this, water moves from petiole of the leaves. Again, here tension is developed. Again, the water moves from the stem region to the petiole. In the stem region, the xylem of the stem, tension is developed. Tension means what? When there is no water present in that region, because the water is moving to the adjacent region, there will be tension developed. This tension causes the pulling of water from the soil. Okay, that's tension. What is cohesion? Now you might feel, is the movement of water taking place against gravitation or along gravitation? Gravitation is this, right? Something which falls down. That's due to gravity. But here water is being pulled against gravity, right? So how come that the water is moving against gravity with such a force? That's because of cohesion. That means water molecules which are present inside the xylem elements, they are all attached to each other by hydrogen bonds. If you remember, usually in chemistry they say that hydrogen bonds are very weak bonds. Then how come hydrogen bonds are helping in movement of molecules against the gravity? That's because though the hydrogen bonds are very weak bonds, but there are many hydrogen bonds. As a result, it forms a strong force. One hydrogen bond is very weak, but there are thousands and millions of such hydrogen bonds which are present inside. As a result, it can withstand the gravitational force. And this cohesion helps in maintaining a continuous water column in the xylem ducts of the plant. So basically that's the reason it is called cohesion tension theory, or it is also called ascent of sap. That means movement of water in the xylem sap. Okay, so this was the theory which was accepted by many scientists and which actually proved how actually water gets transported from the roots till the peaks of leaves in tall plants. Okay, you can see here evaporation is taking place in the leaves, that is causing the tension in the leaf region. You can see how in the xylem tracheids, which are xylem vessels, which are separated by these perforation plates, water is in the form of a column. It is raising in the form of column. That's because of these hydrogen bonds, the strong hydrogen bonds. And this causes the movement of water from the soil to the roots. So there is tension developed in the leaves that causes the pulling. This tension is developed due to transpiration. Clear? Okay, now we have understood how actually transportation takes place, how water which is absorbed by the roots is reaching the leaves which are present at the topmost part in the trees. That's by cohesion tension theory, which was proposed by Dickson and Joly. Now let us understand. Okay, till now we have talked about xylem. We know whenever we are talking about this transport of water, which tissue is playing a key role? It's the xylem. It is the vascular tissue along with the xylem vessels, tracheids and the fibers plays an important role in transport of water along with mineral nutrients. Now let us talk how actually the product of photosynthesis, which is glucose, is getting transported from the leaves to various parts. Okay. Now what is source? It is the region where food is synthesized. Okay. Now leaf, we have studied from our lower classes that leaves are the regions where the food is being produced. So basically by the process of photosynthesis, we know glucose is the end product. Okay. So this glucose has to be transported to various parts of the plant. But one thing you need to remember, the product of photosynthesis is glucose. Okay. But this glucose has to be converted into sucrose, because sucrose is the transported form of glucose. It is the transported form. Sucrose is a disaccharide, means made up of 2 sugar units. It is made up of glucose and fructose. Okay, so glucose has to be converted into sucrose, and this sucrose has to be transported to flowers, roots, stems, the developing buds, etc. Okay, so source is the region where food is synthesized. So the young leaves, they act as good source. And what is sink? It is the region which needs that food. Now a flower, or it can be a developing leaf, young leaf, or it can be the roots, it can be the stems, it can be the petioles, all these, it can be the stem, all these require the food. Okay. And translocation is the mass or bulk transport of food or water through phloem and xylem from the leaves to various parts of the plant. So bulk movement, it can be bulk movement of water, bulk movement of organic nutrients, that is called translocation. If it is food, it takes place through phloem, and if it is water, it takes place through xylem. Now, how actually this movement takes place? To explain that, there is this theory called mass flow theory or pressure flow theory. This theory was put forward by Kramer Russell and Barber. Okay, they proposed this theory, mass flow or pressure flow theory, to explain how actually the transportation is of organic nutrient, that is sucrose, is taking place. I already told you product of photosynthesis is glucose. This glucose is synthesized. Now this is what leaf cell. What is leaf acting as? It is acting like a source. Okay, and root cell is a sink. It needs the food. Okay, and in the leaves we know chloroplasts are present. They are the factories where food is being synthesized. And how is the food synthesized? In the form of glucose. But can glucose be transported? No, the transported form is sucrose. Okay, this sucrose which is present inside the leaf cell, how is it going to the root cell? That was explained through mass flow or pressure flow, which was given by Kramer, Russell and Barber. Okay, and remember, sucrose is a disaccharide and it is made up of, it's a disaccharide which is basically made up of glucose and fructose. It's important, okay? And transported form is sucrose. That's also important. Okay, now see here how actually it happens. See, in the source, that is leaf cell. Now if you see here, this is phloem. These are the sieve cells. Each one is sieve cell. The sieve cells are connected to each other and that forms a sieve tube. The sieve tubes are separated by sieve plates that allows the movement to take place in this way. And you have plasmodesmatal connections, and this is the companion cell. Okay, now this is the source where the sucrose is getting transported. Sucrose gets transported to the phloem parenchyma, and from the phloem parenchyma it reaches the companion cells. From the companion cells, the sucrose moves into sieve cells. Now what actually is happening is, sucrose, is it a solute or a solvent? It is solute, right? So here what's happening? Water potential is decreasing. Why is it decreasing? Because there is more addition of solute in this region, in the phloem. So water potential is less. But beside, you have xylem elements present. If you really cut a root section or leaf section, obviously phloem and xylem are associated together. Okay, they form a vascular bundle. So that's the reason they have drawn the xylem and phloem together. Even inside the cells, the xylem and phloem are present close to each other. Now in the xylem is rich with water, right? It has water and mineral nutrients, so water potential is very high. We know water moves from a region of higher water potential to lower water potential. So what actually happens is, this water starts moving from xylem to phloem, and this causes the movement of water along with these solute molecules to pass into the sink. Okay, so this process, now see here, this is called loading. What is loading? Loading is where the sucrose molecules present inside the source are getting loaded into the phloem, so that they have to be moved into the sink. Okay, now see, source is what? Leaf. What is sink? It is your root. We want the sucrose which is present in the source to reach the root. But can it occur directly? No. Who is helping for that? Phloem cells. Which phloem cells? The companion cells and sieve tube cells majorly, because phloem parenchyma is mostly involved in support and storage. Okay, so basically what happens is, the sucrose, the movement of sucrose into the companion cells and the phloem is called loading, because it is filling the phloem with the sucrose that has to be transported to the sink. Now as the phloem is loaded, its water potential reduces. This causes the water to move from xylem to phloem, and that causes the movement of the solute molecules to the root. Okay, now in the root, all the solute molecules get unloaded. Okay, just imagine like you have ordered a gift or something in Amazon. Then what happens? We know that they pick the product from the shop, or that is called loading, and after that they give it to our home. That's unloading. In the same way, the leaf is the source here. Okay, that sucrose has to be transported to the root, so loading takes place through phloem. Okay, and unloading occurs in the sink. Is it clear? Now, after unloading, what actually happens? Are there any solute molecules present inside the phloem tube? There are less solute molecules present. Again, here water potential increases and the water starts moving back to xylem. In this way, there will be movement of water from xylem to phloem during loading process, and during unloading, when the solute molecules are reducing in the phloem element, the water starts moving back to xylem. Okay, so it is very important to understand this process of loading and unloading. What is loading? The sucrose which is present inside the chloroplast of the source, when it gets loaded into the phloem element, that is called loading. And during loading, water potential decreases because you are adding more amount of sucrose in it. So therefore, water starts moving from xylem. During unloading, water potential starts increasing. As a result, water starts moving from the phloem element to the xylem tubes. Is it clear? So this mass flow or pressure flow hypothesis explains how the food transport takes place from the source to the sink. And one important thing to remember, it is not that always leaf acts as a source. Under unfavorable conditions, sometimes roots also can act as source. In case of potato, for example, if you see the root tubers, how are they? They are very swollen, they are rich in starch. Okay, so sometimes the root tubers provide food to the developing leaves, to the young buds or flowers. Okay, so that's the reason they say it as bidirectional transport. Okay, let us end the topic with mycorrhiza. What actually are mycorrhiza? I'm sorry. It is a symbiotic association between fungus and roots of plants. Okay, a symbiotic relationship. What do I mean by saying symbiotic relationship or association? That means a mutual relationship. Just take an example. You yourself and your friend are thinking to prepare a cake. Both of you are thinking to prepare different flavors of cake. You are thinking to prepare a strawberry cake and she wants to prepare vanilla cake. And you have a oven, everything, but you don't have the baking powder. And even your friend, she's thinking to prepare vanilla cake. She has everything except vanilla extract. So you say that I'll give you vanilla extract, you give me the baking powder. So what's happening? Both of you are exchanging the items that are available with you and start preparing the cake. In the same way, symbiotic association means here both partners help each other, the partners who are involved. One partner is fungi and the other partner is plants. Okay. Now we know that fungi are heterotrophic. What do I mean by saying heterotrophic? That means they cannot prepare their own food. Okay. They depend on plants. And plants are autotrophic. Obviously they can synthesize their own food. That's because of the pigment chlorophyll. So these fungal cells, they form mycelium. What is mycelium? Mycelium are root-like structures. Okay. They form mycelium and they get associated with the roots of the plant. And these fungi, what it does is, it increases the surface area of absorption of water and nutrients for the plant. Okay, it sees that plants can absorb more amount of water from the soil and allows the phosphorus absorption more. And in turn, plants, what they do, they provide nutrients by process of photosynthesis. So plants give food to the fungal mycelium, and fungal mycelium see that more amount of water and nutrients are absorbed by the plant by increasing their surface area of absorption. So it is a symbiotic association. Okay, mycorrhiza is a symbiotic relationship where both partners help each other and they survive together. Okay, where the fungal organism depends on the products of photosynthesis on the plant, and plants depend on fungi for efficient absorption of water and minerals. Another example of symbiotic association is your leguminous plants with rhizobium. In that case, legumes are nothing but beans, pea plants and all, and rhizobium is a bacterium. This rhizobium helps in providing nitrogen to the plants, and plants provide food to the bacterium. Okay, even that's an example of symbiotic association. Okay, so in this unit we have discussed how actually transportation of water and food takes place. We started from the basics, like what actually is a cell, what is plasma membrane, how is protoplasm different from cytoplasm, clear distinction of protoplast and protoplasm, what is a tissue, why xylem and phloem are considered as the complex tissues where they are made up of different types of cells. Water, what is the amount of water present in different cells or different plants, and how much water is actually available to the plant. Is all the water present in the soil available to the plant? No, we have studied it's the capillary water. Then we have discussed the main mechanism, water potential, then units of water potential. Then I explained you the how water actually moves from a region of higher water potential to lower water potential, or we can also say how water moves from lower negative water potential values to more negative water potential values. Then we discussed how movement of water takes place in the roots, the symplastic apoplastic pathway, diffusion, facilitated diffusion, active transport or uphill transport, what is osmosis, hypertonic, isotonic, hypotonic solutions, and how it takes place in animal cell, in plant cell. The main difference is animal cells lack a cell wall and plant cells, they have a rigid cell wall. Then we discussed about imbibition. Later, we talked about the theories. The best theory which explains is the cohesion tension theory. And how translocation takes place. To understand that, first we need to understand what is the source, what is the sink, what is the main transported form of sugar, that's your sucrose. Then we discussed it's the mass flow or pressure flow theory which explains it. Why is it called mass flow? That's because the solute molecules are moving in a bulk. Okay, it's not that a single molecule is moving. Mass flow. And why it's called like a pressure flow? That's because there is less solute potential present in the phloem. Water is moving from xylem to phloem. This causes the pressure for the water, for the solute molecules to move towards the sink. Okay, loading, unloading, and mycorrhiza. Okay, where it's the symbiotic association between fungi and roots of plants. Thank you.